Myself Dr. Vivek Kumar, Professor, Himalayan School of Biosciences, Swami Rama Himalayan University, Jolly Grant Dehradun. I am again with you with a new topic on endophytes, that is the endophyte plant biotic stress alleviation. In plants, the biotic stresses are initiated by organisms, the living organisms. It could be bacteria, it could be fungi, it could be insect, it could be any nematode, or it could be any animal. So, viruses they are also included, spiders, weeds, etc., etc. The biotic stresses they take nutrients away from their host, spread disease, reduce the vigor of the host plant in the severe cases. The biotic stresses lead to pre and post harvest yield losses all over the globe. The plant lacks the adaptive immune system, but they can counter biotic stresses by developing complicated strategies. Now, there are certain resistance mechanisms in plants that perform against biotic stresses. They are regulated innately by plant genetic code and genes against the diverse biotic factors or stresses. The biotic stresses are entirely dissimilar from the abiotic stresses, since abiotic stresses are caused by non-living, while the biotic stresses they are caused by the living beings. So, several biotic stresses influence normal plant physiology and photosynthetic rate, as leaf munching insects reduce leaf surface area, resulting in reduced photosynthesis and the plant growth. The bacterial endophytes they have an advantage over bacteria in inhibiting the rhizo or phyllosphere. Since living within the plant tissues, it represents an opportunity to be in constant contact with the plant cell, and endophytes become more integrated in plant metabolism than rhizo or phyllosporic microorganisms. Therefore, they are able to neutralize the biotic stresses more efficiently as compared to the phyllo or rhizospheric microorganisms. Now, if we discuss the microbial pathogens, the secondary metabolites production by most endophytes have been reported, and some of them display antifungal, antibacterial activities which benefit in preventing the plant pathogenic microbes growth and development. So, diverse bioactive composites they have been reported for the capability to impede the growth of several host pathogens. Similarly, several secondary metabolites of endophytes having endomicrobial potentials have been reported, such as alkaloids, steroids, peptides, quinones, polyketics, and phenols, etc., etc. So, the endophytic microbes have been reported to restrict or remove, delay or prevent the growth of phytopathogens under various agroclimatic conditions. This they do by synthesizing numerous antimicrobial compounds. The literature has more information about Bacillus and the Pseudomonas bacteria as the major bacteria investigated for their potential to manufacture diverse antibiotics, such as pyrrolnitrine, zimcrolactone, ecomycin, urgina, hydrogen cyanide, etc., etc. And various strains of Pseudomonas genes are known for manufacturing the very types of antibiotics that significantly suppress agricultural diseases, such as pyroluteroin and 2,4-diacetyl fluoroglucinol by Pseudomonas fluorescens to overpower Thylaviopsis basicola, which is responsible for tobacco root rot disease. The spray of Bacillus amyloacufacciens on pitaya fruit resulted in a reduction of lesion caused by Alternaria alternata as compared to the control plant. And this picture shows that there are different types of, we can say, biotic stresses, different factors, viruses, bacteria, fungi, insect, nematode, etc., and they cause stress to the plant. But if we use the endophytic fungi, it is acting as a biofertilizer, it is acting as a biocontrol agent, and it is protecting the plants against diverse biotic stresses. And they discuss the insects. The insects they are accountable for an annual loss of about 20 to 36% of the crop production globally, the cost of which is estimated up to $470 billion, and maximum pre-harvest losses of the crop 13 to 16% all over the world. Further, the post-harvest crop storage loss by pest infestation also constitutes another loss of agricultural products, about 50 to 60% of stored grains, due to inadequate storage and control measures. Entomopathogenic fungi they play diverse roles in nature, comprising endophytic plant growth stimulation, pathogen inhibition, and rhizospheric colonization. Entomopathogenic fungi, they reduce plant diseases by using several mechanisms. The most common approach is the pest development rate retardation, insect feeding inhibition, insect larval reduction, survival reduction, and insect reproduction rate reduction. Beauveria bassiana and Metarhizium, they efficiently control citrus rust mite under greenhouse conditions. Treatment with conidia concentrations of 1 into 10⁸ ml per liter of Beauveria bassiana resulted in more than 60% mortality of Phyllophthora olivora under greenhouse conditions. Vitis vinifera treatment with 3 strains of entomopathogenic fungi Metarhizium resulted in the mortality of larvae 75%, pupae and adult up to 85%, and this we can say Lobessa botrana, it is a severe pest which has been controlled in Vitis vinifera using Metarhizium. Bacillus thuringiensis endophytic isolates BRB1, VLG15 isolated from legume root nodules having cry1 and cry2 family genes. So, this picture shows the Bihar hairy caterpillar, and the Bacillus thuringiensis have been reported to control the growth of this Bihar hairy caterpillar growth. Now, the nematodes, the use of we can say endophytes as a biocontrol agent against Melodogyne incognita have been widely employed in tomato crop. The bacterial endophytes Bacillus widmani, Serratia liquefaciens, inoculation to tomato plants resulted in reduced egg mass and galls of nematode Melodogyne incognita. Bacterial endophytes Pentoa agomirans and Pseudomonas putida, inoculation with ornamental plants in pot experiment resulted in reduced gall index of Melodogyne incognita as compared to the uninoculated control plants. Again, we have seen this picture in the we can say the abiotic stasis. If we discuss the biotic stasis, these are different biotic stasis: bacterial pathogens, fungal pathogens, viral pathogens, insects and nematodes. So, these all living organisms, they act as the biotic stasis, and these can be mitigated, controlled, managed by the different enzymes, antibiotics and chemical secondary metabolites produced by the endophytes. So, in a post experiment inoculated tomato with Bacillus cereus and Bacillus cereus another 2 strain and Bacillus altitudinus, the 3 Bacillus strains inhibited the Melodogyne incognita egg mass up to 81% and gall number up to 75%. Bacillus thuringiensis strain AK08 inoculation to the tomato plants resulted in mortality up to 95 and 4.6% of Melodogyne species. Strain also produce other ancillary and metabolic compounds such as steroids, flavonoids, phenols, alkaloids, tannins, etc. And the main compound detected in endophyte with GCMS assay was clozate 5N3-ol carbonochloridate, which might be considered as having the nematicidal activities. The several endophytic bacteria, Enterobacter, Serratia, Bacillus, when isolated from Robusta coffee, they exhibited nematicidal activity. Out of these, Streptomyces demonstrated noteworthy nematicidal potential against Melodogyne, which inhibited hatching eggs 85%, caused mortality 85%, juveniles of J2 stage. Serratia strains have been reported to produce ceratomolite like lipopeptides, showing mortality up to 77% of Bursa philancus xylophilus, which is the pinewood nematode. Diverse biotic stasis like pathogenic microbes, harmful insects and nematodes no doubt have a seizable unhealthy impact on the plant, such as less photosynthesis, poor plant growth, poor root development and root development. However, this effect can be alleviated by use of the endophytes. The culture filtrate of endophytic Fusorum solani showed 100% juvenile mortality of Melodogyne javanica after 48 hours under in vitro conditions. Endophytic fungi, Beauveria bassiana, Piscillomyces, Metarhizium in coffee crop acted as a wonderful biocontrolling agents against nematode. Similarly, viruses, the viral disease and their damage causing significant loss to the economic crops. Virus cause major crop disease. So, one of the possible measures to control virus is the use of ribonuclease RNAs, which can cleave the RNA in the viral genome. So, Bacillus strains with high ability to colonize internal plant tissues they combined with high RNase activity reduce severity of the viral disease symptoms in the plants and in the potato virus. The ability of Bacillus pumilus, Bacillus amyloaquiescens, Bacillus licheniformis to produce extracellular RNases like bioanases, the different types of RNases. So, low concentrations of RNases stimulate plant growth and resistance to broad spectrum stress factors, and high level of them show antiviral properties by destroying the viral RNA. Thus, in one investigation, Alpentoia, Chronobacter, other bacteria which originate from Cucurbitaceae plant produce nucleases, and these nucleases killed the genome of the virus. Finally, we discuss the keywords. So, the beauty of an endophyte is its functional role in improving, enhancing the plant growth and defending it from different abiotic and biotic stresses. Abiotic stresses and biotic stresses, they are entirely different, and once they both come together, then the health of the host plant is severely affected. So, this attribute does not encompass host specificity. Therefore, one can utilize endophytes as bioinoculants to mitigate diverse stresses resulting from changing ecological forms. And with growing attention on food security, ecosystem protection and sustainable agriculture, so the beneficial endophytes exploitation is crucial. Beneficial endophytes exploitation means we have to identify and we have to screen certain endophytes which are able to really enhance the growth of their host plant, which are really protecting the health of their host plant by using diverse means. The diverse means which we have discussed in this lecture as well as in the previous lecture. See you in the next lecture. Thank you so much.